everyone, welcome back to ConceptWala. In our previous video session, we were talking about the basic structure and function of MHC molecules. In this video session, we are going to talk about the exogenous pathway of antigen processing. Now see, the immune cells of our body always try to eliminate the pathogen at the very first place. So when an antigen enters in our body, the antigen presenting cell internalize the antigen through endocytosis. Now let's just learn the whole process through diagram. It will be much easier to understand. So assume that this is the membrane of your antigen presenting cell, and now for example, let's just say the macrophage. Okay, so now this macrophage has internalized an antigen through endocytosis. Now this is your endocytic vesicle and this is your antigen. Okay, now this antigen will move forward through some increasingly acidic endocytic vesicle. Now you guys know that this endocytic vesicle is filled with hydrolytic enzymes, and these hydrolytic enzymes will degrade the antigen in oligopeptides of 13 to 18 amino acid. Okay, so now this was your early endocytic vesicle and this is your late endocytic vesicle, and now the hydrolytic enzymes has degraded the antigen in oligopeptides of 13 to 18 amino acid. So these are the oligopeptides. So now let's just hold it here. Okay, so you guys know that the MHC1 and MHC2, both these MHCs are synthesized in rough endoplasmic reticulum. So just assume that this is the region of your rough endoplasmic reticulum and this is your MHC2 molecule. Okay, now in this rough endoplasmic reticulum, alongside this MHC2 molecule, another protein molecule which is known as invariant chain is also synthesized. Now this invariant chain will occupy the peptide binding cleft of MHC2 molecule. Okay, so this is your invariant chain in the green color and it has occupied the peptide binding cleft of this MHC2 molecule. Now this invariant chain basically prevents any premature antigen binding to this MHC2 molecule, and it also has sorting signal in its cytoplasmic tail. Now this sorting signal will direct the transport of this MHC2 molecule to endocytic vesicle. Okay, so now this is your endocytic vesicle and this is your MHC2 molecule. And now in this endocytic vesicle, the hydrolytic enzyme will degrade the majority of this invariant chain, but a very short fragment or a short portion of this invariant chain, which is known as clip, will remain attached with the peptide binding cleft. Now this is your clip, the short fragment of this invariant chain. The hydrolytic enzymes are not enough to remove or to degrade the clip portion. Okay, so here comes another molecule which is basically a non-classical MHC molecule that is known as HLA-DM. That HLA-DM will catalyze the exchange of this clip with the antigen. Okay, so this is your HLA-DM. Now this HLA-DM will catalyze the exchange. So basically now these two endocytic vesicles will fuse. So these endocytic vesicles has fused and this is your MHC2 molecule and this is the antigen. The HLA-DM has catalyzed the exchange. Okay, so now this endocytic vesicle, which is basically known as M2C, if I am not wrong, this is known as M2C. So this M2C will fuse with the plasma membrane and the MHC2 molecule with the antigen will reach the plasma membrane. So now this is your MHC2 molecule and this is your antigen. Now CD4 plus T cell will come and recognize the antigen and it will proceed with the immune responses. Okay, whatever immune response is necessary for this antigen. Now this was the whole thing about the exogenous pathway. It is very easy. In our next video session, we are going to talk about the endogenous pathway. So you guys can download the PDF of this video session or PDF of the lecture from our website, which is www.conceptwala.com, or I will put the link in the description box. And the next video session will be the last video session of our MHC series. So till then, just keep watching Concept Wala and keep learning and keep growing.